To understand how microbes can be applied to biotechnology, it is first important to understand what biotechnology is. Biotechnology is the use of microorganisms or living systems to produce or develop products. I will first be talking about Aspergillus oryzae in biotechnology. So from the experiments that we carried out, this fungal's resistance to antibiotics in optimum growth conditions and varying concentrations of glucose can be seen. Aspergillus oryzae is used in the production of soy sauce to ferment soybeans. Genetically modified Aspergillus oryzae can also be used in large-scale production of lipases to be used in biological washing powders, and fermentation of genetically modified Aspergillus oryzae is a major source of chymogen, which is used in cheese production. Carrying out experiments such as this one allows for the identification of specific microbes' properties that can be implemented to help produce various products. Some of these properties can include rate of growth factors, enzymes, and byproducts secreted, among others. One reason Aspergillus oryzae is used commonly is that it forms condyspores, which allows for the production and distribution of the microbe while still keeping it alive and uncontaminated. This fungal also does not produce any carcinogenic metabolites like Aspergillus flavus does. Both of these fungi are similar genetically but have this major difference. Because it doesn't produce any harmful toxins, it is therefore safe to use in the production of food. Aspergillus oryzae does produce the enzyme amylase, which is beneficial in the human body as it aids in digestion. Because of this, it is an added bonus in the production of food such as soy sauce, miso, and rice vinegars. These are just some examples for one type of fungus. There are countless other species of fungi that are commonly used in biotechnology. Saccharomyces cerevisiae is used to ferment sugar into alcohol and carbon dioxide. Aspergillus niger is used in the commercial production of citric acid. In general, fungi are important in the nitrogen and carbon cycles and the breaking down of dead organic material known as decomposition, which allows for nutrients to be cycled through the ecosystem. Various drugs such as the antibiotic penicillin and cyclosporin A, which prevents organ rejection after a transplant, all derive from fungi. Even some cancer research is being carried out using fungi. From the experiment that tested different antibiotics on Aspergillus oryzae, the effectiveness of varying antibiotics can be seen. This knowledge can be used to determine the best medication for a person who has been infected with the fungal. However, Aspergillus oryzae is not harmful to humans, but the same principle can be applied to other more harmful fungi and other microorganisms that cause disease. It's not just fungi that can be used in biotechnology. Viruses can also be utilised. One of the main pieces of biotechnology that uses viruses is vaccines. When our body has been infected with an antigen, lymphocytes, which is a type of white blood cell, will produce antibodies which fight and kill the antigen. However, the first time the body faces an antigen, the body can take more than several days to give the antibody response, and if the infection is from a more severe antigen, the infection can spread and kill the person before the immune system can fight back. Vaccines are made of dead or weakened viruses. They cannot cause an infection due to their dead or modified state, but the immune system still recognises them and produces antibodies to fight them. Memory cells will be shown how the antigen was fought, and therefore, if the body encounters the antigen again, the body can respond much quicker and more effectively. Essentially, viruses are modified to teach the body how to fight a specific infection. Another very novel form of biotechnology that makes use of viruses is phage therapy. A bacteriophage is a virus that only kills bacterial cells. These phages need a host to survive and reproduce, as they are essentially genetic material in a shell. Usually, a phage targets one species of bacteria or some of their close relatives. They kill the bacteria using the lytic cycle, which is also how they reproduce, and in novel research we have been injecting these phages into our body to see if they can act as an alternative to antibiotics. With the rise of antibiotic resistance, we desperately need an alternative before they are no longer effective, due to the fact that bacteria have become more and more resistant to these drugs. Injecting these phages into our body may be able to solve our problem. Phages will not affect human cells, only bacterial cells, and while antibiotics kill good and bad bacteria in the body, phages are very specific and only attack what they're supposed to. Bacteria will not become resistant to the bacteriophages because as the bacteria evolves, so does the phages, and as the bacteria's phage resistance increases, the antibiotic resistance of the bacteria decreases, giving up one for the other. So we may be able to turn back to antibiotics if needed. This therapy has not yet been commercialised, but more and more research is being put into this type of therapy, and so far it looks promising. Bacteria can also be utilised in many ways in biotechnology. Some of these include using these microorganisms to break down waste, clean up oil spills, produce biofuels, create antibiotics, create important proteins such as insulin, in probiotics, and in the production of cheese and yogurt. A specific example that there has been a lot of research into is bacteria being used to clean up oil spills. Oil spills are a major problem as they cause catastrophic damage to the local ecosystems in a number of ways. The bacterium Alkanovorax borgumensis is naturally found in the ocean but is quite rare. It relies on oil as an energy source, and it is a set of genes that allow for it to break down the alkanes in the oil to use them as food. They can also take advantage of the hydrocarbons found in the oil, and it can make use of both organic and inorganic nitrogen found in the oil. Researchers have been sequencing the genome of this microbe to harness its features that allow it to use oil so future oil spills can effectively be cleaned up. It is not just microorganisms that can be used in biotechnology. There are many other types of biotechnology that use other living systems such as in vitro fertilization, gene therapy, transgenics, and clustered regularly interspaced short palindromic repeats, or CRISPR. Other examples include research into various animals to see if their features could be applied to other circumstances. For example, zebrafish can recreate brain material, unlike humans. Discovering what genes or how this fish does so could lead to potential cures for neurodegenerative diseases and brain damage. And because 70% of the protein-coding human genes are related to genes found in the zebrafish, there is a chance that this feature could be applied to humans. Another animal that is a form of biotechnology is mice. Mice are the most commonly used research animal as they act as the model organism for the function of mammal systems, which means they can be utilised to study various factors such as diseases and treatments and how they may affect mammals, including humans. Gene therapy is a very novel treatment that a lot of research has been going into. It is a technique that uses genes to treat or prevent disease. It involves inserting a gene into a patient's cells, and currently it is being researched that a mutated gene that causes disease such as cancer could be replaced with a healthy gene, or in fact in activating a mutated gene that isn't functioning properly, or in introducing an entirely new gene to help the body fight disease. This has many potentials for treating inherited disorders, cancer, and some viral infections. However, the safety of this treatment, and it is currently only being tested in patients who have diseases that can be cured in no other way. It can clearly be seen that there are many forms of biotechnology, from fermentation to gene therapy, and knowledge from these experiments, such as the growth conditions of fungi, can be used to discover the potential of living systems in creating and developing products where it can be for medicine, food, or the environment.